Hello everyone and welcome to today's webinar, Next Generation CMOS Cameras for Neuroscience Research. I'm Cassie Saltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Teledyne Photometrics. To learn more, visit www.photometrics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Matthew Kose-Dunn, Content Manager at Teledyne Photometrics. Matt, you may now begin your presentation.
Brilliant. Thank you, Cassie.
Hello and welcome to another Teledyne Photometrics webinar with me, Dr. Matthew Kose-Dunn. This new webinar focuses on neuroscience and ties into Teledyne's upcoming presence at the 2023 meeting for the Society for Neuroscience, running from November 11th to 15th this year. If you want to come along and have a chat in person or learn more about the latest in modern camera technologies, come over to SFN this year. Firstly, a little bit about Teledyne Photometrics. We design and manufacture high-end cameras for scientific research, everything from imaging cells to imaging space. Our products and solutions support demanding quantitative research no matter your application. We're the market leader for scientific CMOS camera technologies and have over 40 years of experience, being established in 1978 and part of the Teledyne Imaging Group since 2019. If you want to learn more about cameras, microscopy, imaging and more, swing by our website on www.photometrics.com. As mentioned earlier, Teledyne will be at SFN this year from November 11th to 15th in Washington. Not only can you see Photometrics, we're coming alongside two other Teledyne business units, namely Princeton Instruments and Luminera. Between us, we represent the whole breadth of imaging, microscopy and spectroscopy sciences, meaning there will be something for everyone. Now, on to the webinar. Neuroscience is a vast and varied field of research focusing on the nervous system. If we look at humans for an example of the nervous system, we will see a highly complex and dense network of nerves that serve a wide range of purposes, everything from locomotion to sensing the world around us. This nervous system can broadly be split into the central nervous system, which consists of the brain and the spine, and the peripheral nervous system, which consists of nerves in the periphery of the body, such as the limbs. Beyond this, there is also the autonomic and enteric nervous systems. The list goes on. These sections are dense and complex and highly challenging to study. The takeaway here is that while it is vital to understand both the structure and the function of the human nervous system, it is a highly challenging area to research, and I say this as a neuroscientist myself. This also means that imaging in neuroscience can be equally challenging, but you'll see that our cameras are up to the challenge. Something to note in neuroscience is the prolific usage of model organisms. Access to a functioning human nervous system is very challenging to get, either a physiologically healthy one or one affected with neurodegenerative or pathological conditions. However, due to evolutionary conservation, we can often glean relevant information by studying other, less complex organisms. Classic model organisms include zebrafish, mice, fruit flies, and nematode worms. Nervous systems of these organisms can be entirely mapped and understood and even imaged in their entirety thanks to their smaller size. Data from these studies can often be applied across the board, revealing structural and functional information that can be relevant to all organisms, influencing our thoughts on mechanisms behind the human nervous system as well. Many neuroscience studies, regardless of the organism used, rely entirely on imaging and microscopy in order to gather data. However, due to complex hierarchical structural networks and high-speed dynamic millisecond scale functional activity, imaging in neuroscience presents unique challenges. Cameras used in these imaging systems must be flexible and powerful in order to meet the demand and answer necessary scientific questions. Neuroscience demands a lot from cameras. Cameras should have a large field of view in order to image as much as a neuronal network as possible and to make experiments more efficient for high content imaging. Cameras should have a high sensitivity in order to capture weak signals over noise, especially at low exposure times. High resolution is also necessary in order to pick out single neurons from dense networks or to trace minute axons and connections. And lastly, for functional studies, cameras need to be extremely fast in order to capture functional activity, whether imaging calcium ions or the voltage activity itself. Despite the numerous challenges of these applications, Teledyne Photometrics offers ideal solutions. Let's go through some of these applications now. Firstly is structural imaging, namely imaging of neural cells or tissues to observe cell morphology, organization, density, and more. This involves imaging across scale, all the way from a single neuron up to large 3D tissue sections or neural organoids. Most important here is to have a flexible solution that can image high enough resolutions to perceive subcellular structures while imaging thousands of cells at once. This often requires cameras with a large field of view and a small pixel matched to the magnification of the imaging system. Here we have an example of structural imaging with the kinetics from Tom Lumen of ETH Zurich. He is using a spinning disk system for multi-channel imaging of mouse brain slices at 20x magnification. Not only did Dr. Lumen need at least 25mm field of view to match with the Crest Optics X-Lite V3 spinning disk system, as well as a small pixel to match with the 20x magnification, he also needed high acquisition speeds in order to synchronize the sCMOS rolling shutter with the disk rotation speed. By using two kinetics cameras that are each imaging two channels simultaneously, this system can image 4 wavelengths simultaneously across a very large area, easily able to sequentially image across an entire mouse brain slice in a high throughput manner, while retaining subcellular resolution. As Dr. Lumen says, they didn't have to compromise, and that's what the kinetics camera is about. Next up are several functional applications, starting with optogenetics. Optogenetics involves using light to control neuronal function. It's a groundbreaking technique that allows researchers to switch individual neurons or whole populations of neurons on and off with just light. It's an excellent technique for exploring brain function. There are several steps to enabling such control. Firstly, a light-reactive protein needs to be identified. These are typically opsins, such as channel rhodopsin or bacteria rhodopsin. The gene responsible for producing this protein is isolated and transfected into sample neural cells using a viral vector, such as a lentivirus. By combining the gene for this opsin with certain ion channels on a neuron, essentially light can be used to force a conformational change in the opsin proteins and activate the ion channels, which results in neural activation, inhibition, or even modulation. Essentially, neurons can be turned on and off at the flick of a switch using light. This can be done with genetic modification of entire organisms such as mice, who can then have a window installed in their cranium to shine light directly onto the cortex and change their neural activity in certain areas while the animals are awake and behaving. And this gives great insight into function. You can control specific individual single neurons or entire brain regions and networks. A great example of this comes from Dr. Zilbertal of Stanford University, who is using optogenetics to image functional neural activity across an entire cortex using a transparent window. This requires a high sensitivity, high speed camera that can image changes in functional activity due to combined fluorescent markers, and the kinetics is an ideal solution to this application. Dr. Zilbertal and team are enjoying the improvements in resolution, field of view, and speed with the kinetics compared to typical sCMOS cameras. Another major technique for imaging neural activity is calcium imaging. The calcium ion, Ca2+, is highly relevant for neural function, and measurement of calcium ion transport is an indirect way to measure neural activity. By using fluorescent calcium indicators, functional activity can be identified and quantified by looking at the signal intensity. These calcium indicators are typically either chemical dyes such as FURA2 or genetically encoded calcium indicators with fluorescent proteins such as GCAMP. As you can see from these 4 examples, by using fluorescence microscopy we can measure calcium activity and therefore function using our cameras. A good example of this is a combination of calcium imaging and light sheet across an entire zebrafish larvae brain from Dr. Isaac Bianco of University College London. This application needed a very large field of view and a high-speed camera to capture calcium activity across a large 3D volume, where fluorescence activity is captured across multiple Z-stack slices. This combines the demands of both light sheet microscopy and calcium imaging, where the former needs you to image across a large area, often with a low light dose, and the latter needs you to image at a high speed. The kinetics is again ideal for this research, as it's also widely used in light sheet applications. While calcium imaging measures calcium ion transport as an indirect measure of neural activity, the electrical activity itself within neurons can be measured directly. This is known as voltage imaging. Similarly to calcium imaging, this requires fluorescent voltage-sensitive indicators, either chemical dyes or genetically encoded voltage indicators, GEVIs, which are combined with fluorescent proteins. Unlike calcium imaging, which is often done at video rate imaging of around 100 frames a second or 100 Hz, voltage imaging occurs on a much faster timescale, on the millisecond timescale. Neuronal activity is extremely fast, and in order to image this, a camera needs to capture fluorescent activity with at least 1,000 frames a second or 1 kHz. In order to have a camera image this fast, exposure times need to be very low, at least 1 millisecond or below. So in order to image at such a high speed, a camera needs a very low exposure time, which means only the most sensitive cameras are able to capture signals at this speed. There's no point imaging at a high speed if you can't capture the desired signal. Voltage imaging is a highly challenging technique that demands the most sensitivity and speed from a camera, but it allows researchers to look directly at voltage activity within the membranes of neurons. The kinetics is an ideal fit for voltage imaging due to its combination of sensitivity and speed, as attested by Professor Xu Han of Boston University. Professor Han has developed GEVI markers such as SOM-Archon and needed a camera capable of very high speeds. Here is an excerpt from a member of her lab with regards to the kinetics' performance.
This is in vivo superficial cortical neuron in a awake mouse. So the mouse is basically on a ball running and doing his thing. This is really in vivo active animal. This is when we first got the camera, did this basic test using a 40x objective in a mouse where we have access to the visual cortex, and we just took full field. And in this case, we had like a small laser spot which was focusing on one neuron. So what we wanted to test is, can we record the full field while testing whether we can see the membrane voltage of a single neuron. As you can see to the right, we were able to see the spikes, which showed that this 8-bit mode does work, because this was the question whether we can see high signal to noise spikes and at least with 40x it's definitely possible, and I think this is pretty awesome. The most surprising for us was that we were able to record almost basically full field at 500 Hertz, and we were able to see single spikes and a good signal to noise membrane voltage of the neurons. So that's very, very promising. Also compared to other cameras we tested, this is only the first camera that was able to do this. And this were able to record many cells at once, or to record few cells at very, very high sampling rates. So this opens a lot of new opportunities. Yeah. Quality of the signal was definitely also something which surprised us compared to other cameras we had. And I think with the kinetics camera, we can record basically at full field of view. So we will be able to record many more, like probably hundreds of neurons. My impression looking on the data collected, the signal to noise seems to be similar to what we know from single cell recordings. This makes me really hopeful that we will be able to record from hundreds of neurons at very high quality. So that will definitely be a game changer in the field.
If you want to learn more about voltage imaging, we have a previous webinar on this topic available on our website. In summary, neuroscience demands a lot from cameras. Large field of view, high sensitivity, high resolution, and extreme speeds. Here, we outline our offerings for neuroscience, starting with the kinetics camera. The kinetics is the next generation of scientific CMOS cameras, featuring extreme speeds across a large sensor, all with very high sensitivity. When you compare it to a typical CMOS camera, the kinetics has over 8 times the speed, an imaging area over 2.5 times larger, and less than 1 electron of read noise. There really is no need to compromise, and it makes the kinetics the ultimate solution for neuroscience, imaging everything from whole organisms to dynamic voltage activity. The kinetics has seen widespread use across the world for neuroscience research and has shown its ability to improve on previous solutions every time. To look a little bit closer at the specifications of the kinetics, we have a 29mm field of view. This is much larger than typical EMCCDs or even typical CMOS cameras, and with a larger field of view, you can capture more information in every single frame. This gives you a much higher throughput, and it can maximize the amount of information that your microscope is able to show you. If you're imaging very large samples, and you often need to spend time tiling and stitching, with a larger field of view, your experiments become much faster, your samples are subject to less light dose, and you get a lot of efficiency. Or, as we saw with the simultaneous multi-channel imaging, you can pair kinetics with a splitter. You can split the large sensor up to 4 times to image multiple focal planes, multiple channels, even multiple polarities, all to give you more information. We also offer the Kinetics 22, which is a 22mm camera optimized for microscopes or systems with a smaller maximum field of view. But in general, the Kinetics has one of the largest fields of view available for scientific CMOS cameras. Here we have a comparison of what it's like to image larger samples when you need multiple captures. With a typical CMOS, giving yourself around a 5 to 15% overlap, imaging across multiple wavelengths and multiple Z-stack field of view, a typical CMOS would take around 25 acquisitions. Considering each picture needs to take multiple wavelengths and scan through multiple Z-stacks, this can take a very long time. With the Kinetics, you can acquire this same image over 3 times faster with just 9 acquisitions, because every single acquisition you take is a much larger field. So in this manner, the Kinetics can really give you a lot of efficiency back with high throughput imaging thanks to its larger field of view. Here's another great example of this from Dr. Ulrich Kubitschek from the University of Bonn. He was doing expansion light sheet where you can physically enlarge your samples, and here you have dense neuronal tissues. You can see with a typical CMOS you have to take over 10 tiles of this, but with the Kinetics he only took 2 tiles. Each of these images still contains detailed subcellular information about each of these cells. So the large center of the Kinetics greatly reduces the number of required image tiles while retaining an excellent signal-to-noise ratio, and this allows you to scan across these large, dense neural samples much faster and save them from any sort of photo bleaching or damage and get the whole thing done much faster. The other aspect of great importance is the speed. The Kinetics will image at 500 frames a second across this full 29mm field of view in the speed mode. If we reduce it to a typical 18mm field of view, it goes over 800 frames a second, which is over 8 times faster than a typical CMOS camera. With CMOS technologies, when you reduce the field of view, you can increase the speed. This means that by reducing the number of rows that your camera is looking at, you can achieve extreme speeds. So across half the sensor, you get 1,000 frames, and then 2,000 and 4,000, up to hundreds of thousands of frames a second, over 100 kilohertz at small fields of view. Because it's a CMOS camera, you always get the full width of the sensor, but when you change the number of rows or the height, you can get extreme speeds. This really allows you to see what you're missing in your sample, and it's only possible due to the very high sensitivity of the camera. And this is all happening in the speed mode, which is an 8-bit mode. And the nature of an 8-bit mode is that files produced by this imaging mode are actually half the file size, which is great when you've taken large stacks at extremely high speeds. If we look at a typical CMOS camera at 500 frames a second, you would capture around this much field of view. Whereas with the kinetics, you capture this much. So not only do you have a very high speed, you have this high speed across an 11 times greater area. So again, you get the best of both worlds. You get a large field of view and you get a high speed to image across it. With the kinetics, there's no need to compromise. Since its initial release in 2020, we've been continually updating the kinetics with new features and improving performance. There's been several region of interest or ROI features added, such as multi-ROI or fast ROI switching. If you want to image objects in certain parts of the sensor without having to image the space in between, you can, and this is what we're focusing on with this region of interest features. We also added frame combination features, such as frame averaging and frame summing. These are calculations that the kinetics can do on the fly, where you can add multiple frames together and take an average, or you can sum multiple frames together as well. This is very useful for certain techniques, and because the kinetics has a powerful FPGA, it can do this analysis on the fly. And because we're constantly adding more features to the kinetics, we're also going to have things like global reset and line scan and much more in the future. So watch this space. And if you do have a kinetics, you're able to send the camera into us for a firmware update, and you'll be able to access all of these features yourself as well. The kinetics isn't the only camera with great neuroscience applications. We also will talk about the Prime BSI Express and the Iris 15 cameras. The Prime BSI Express is a powerful, compact CMOS which images high speeds, low noise, all with a very compact form factor. So you get 95% quantum efficiency, 95 frames a second, and 1 electron of read noise. The Prime BSI Express is widely used for neuroscience applications because, just like the kinetics, you get a combination of sensitivity and speed, which is great for certain applications. Overall, the BSI Express is much smaller than the kinetics, so it's easily integrated into existing systems or OEM devices, and it's also widely used for other applications like light sheet microscopy. Overall, it's an ideal solution for the majority of imaging applications, and if you do require greater speed or a larger sensor, the kinetics line is always available. Here's a great example of the BSI Express being used to image an entire zebrafish that's been cleared, so it's been made optically transparent. Because the BSI Express has a small pixel, it's ideal for this kind of high-resolution light sheet imaging, and as you can see, it captures the entire fish brain and more. So the small pixel size of the Prime BSI Express, combined with a relatively large sensor and high speed, is a big advantage, as Dr. Franziska Kurt of the University of Oldenburg tells us. The other camera to mention here is the Iris, and this is designed for very high-resolution imaging. So the Iris has a 25mm field of view, but has a very small 4.25 micron pixel. This gives it 15 megapixels. So the combination of a very large sensor and a very small pixel gives you very high resolutions when you're imaging at low magnification. So you can image large samples at a low magnification and retain a high resolution. And this is ideal for imaging very large, dense samples such as neuroscience samples, cells, tissues, even entire organs. As you can see, it's also very compact, but the main aim here is to achieve subcellular resolutions at low magnifications. Similarly, the Iris 15 is also widely used for light sheet, and light sheet is also a great technique for neuroscience samples. As well as the Iris 9, we also offer a 9 megapixel model, the Iris 9. A great example of the Iris 15 at work is from the owner of the Mesospin project, Dr. Fabian Voigt from the University of Zurich. The Mesospin project is aimed at imaging things at the mesoscale, so very large samples. The large sensor and small pixel of the Iris 15 is ideal for Mesospin, and as you can see here, we've imaged an entire cleared mouse brain at very high resolution. The Iris 15 with its large detector and rectangular sensor are ideal for these kinds of applications. Overall, we have the extreme speeds and size of the kinetics, the balanced combination of sensitivity and speed in the Prime BSI Express, and the high resolution small pixel Iris 15. No matter what your application or sample, there's a camera here that's going to be a great solution for neuroscience research questions. Just to break down in terms of specifications, here we can see the noise and QE for each of these cameras. Again, the Kinetics and Prime BSI Express cameras are back illuminated, so you get a very high peak quantum efficiency in the high 90s, all with 1 electron or less of read noise. These cameras all have a small pixel around 6.5 micron, with the Iris 15 having an even smaller pixel at 4.25, and they all have a large pixel array able to image across a large field at a high resolution. This results in a big field of view overall, and all these cameras can image at a high speed. But as you can see, if you want the best of each of these specifications, the Kinetics family is where you don't need to compromise and you can get the sensitivity and the resolution and the speed. In summary, neuroscience is a vast, complex, and continually developing field, and improvements in camera technology allow researchers to see even more than before, from entire brains to individual neurons firing in real time. This has allowed researchers to get both structural and functional research answers, which have benefited from pairing a suitable camera with a suitable imaging system. And on top of this all is the Kinetics. If you really need to see more of your sample in terms of spatial resolution, field of view, or a temporal resolution in terms of speed, the Kinetics really does not compromise on any of these specifications. And so if you need to see more from your neuroscience sample, the Kinetics line is always here to deliver. I'd just like to say again one more time, Teledyne will be at Society for Neuroscience 2023. Come by booth 2914 if you'd like to see us. I'm going to be there, so if you'd like to have a chat, swing by. It'll be great to see all of you. That's been all for this webinar. Thank you so much for listening, and I'll take any questions now.
Thank you, Matt, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen and click Submit. We'll answer as many questions as we have time for today. Let's go ahead and get started. Our first question that we have asks, what other new modes are coming for the Kinetics?
Yeah, so as I said, the Kinetics, it was released in 2020 and now in 2023, because of the onboard FPGA, we can keep adding new modes to it. So I mentioned multi-ROI, which lets you set multiple regions of interest anywhere you want on the sensor, and then you can do fast switching between them. We added the frame settings, which is to say you can do frame averaging of multiple frames, and you can also do frame summing. One of them is on board the camera and the other one is on the driver. And then we also have line scan, which is where if you get a really small region of interest, you can almost exponentially increase the speed. Beyond those, I would say the next one to come is likely to be global reset. This is also coming to the Prime BSI Express camera that I was talking about. But thanks to the powerful onboard FPGAs, we can essentially make feasibly anything that people would want, as long as it's within the power of the camera to perform. So we have programmable scan mode where you can control the direction and the speed of the readout. Why not have multiple programmable scan modes, like multiple regions and this kind of thing? So for now, I think the next one will be a form of global reset. These are constantly being added to the camera, and so we can do firmware updates to add them to existing Kinetics if you have them.
Wonderful, thank you. All right, our next question asks, pixel size versus object size correlates?
Yes, so the pixel size of this camera, for example, would be 6.5 microns. And then you would be multiplying that through the objective you're using. So if you're using a 10x objective, it would be 0.65 micron at the sample. And ideally, you want to match it for Nyquist, so you want it to be around 100 nanometers at the sample. Because this is assuming you're working with cells and biological samples. If you're imaging cells, you want something that's going to be capable of seeing subcellular details, but also having a large enough field that you can image hundreds of cells at a time. So in general, we offer 6.5 microns as a nice balanced pixel. This is optimized for Nyquist at 60x magnification, because most of these kind of imaging setups have a 10, 40, 60, or 100x. If you're doing 60 or 40, then 6.5 micron pixels is great. If you're doing really high magnification in the hundreds to 150s, then you can bin a 6.5 micron pixel to make it bigger, around 13 micron, or you can use an 11 micron pixel because that's optimized for Nyquist. And at the other end, if you're doing really low magnification, for example, 20, 10, 4x even, then a smaller pixel would also be optimized for Nyquist here. And we do have the Iris with the 4.5 micron pixel. So you can bin the pixels to get to the optimal size that you need to, but it's best to have a camera with the pixel size that's correct for the work that you're going to be doing with it. But largely, we find that 6.5 is a nice balance between them.
Fantastic, thank you. Our next question here says, the online calculator for the kinetics shows that a number of rows below 8 FPS, the frames per second can get up to 1.6 FPS for one row at 8 bits. Are these values realistic?
Yes, so great question. I would say once you get below 100 rows on the kinetics, you're going to want to do some testing, because when you get to about 32 rows, the calculator on the kinetics page says 50,000 frames. And that is what the camera will do, because it does 500 at the full frame, and every time you half the field, you double the speed linearly. So half of it does 1,000. And by the time you get down to 32 rows, it's 100 times the speed. So it's 50,000 frames a second. As you get below that, you start to run into other bottlenecks, which is to say, how fast can the cable transmit data to your computer, and how quickly can your hard drive read and write that data as it's being pushed onto the system. Because there's no point imaging at hundreds of thousands of frames a second if you can't then save these images to the computer without any dropped frames, because you're going to be imaging something very quickly. You don't want any information to be lost. So we recommend, once you get below around 32 frames, you get sort of the single digits, it's best to run your own tests. The array size calculator on the website will just calculate these linearly as to what they should be in theory, but we find that theory and practice is different depending on the computer system you have. If you have a kinetics, it's vital to check the microscope, that you can have a big enough field of view to maximize your microscope. And then it's good to check your computer to see if your computer can accept the amount of data you're pushing down the cable and save it. You can stream to RAM and you can stream to disk. I should say that on the kinetics page as well, there's also a link to recommended PC specifications. We recommend some systems which will give you the full output of the kinetics. And it's good to do some testing with the individual model that you have to see what speed you will get once you get below 5 rows, for example.
Great, thank you for that. Our next question we have here, this person is asking, will this webinar be available to watch later on your website?
Yeah, so it'll be available on LabRoots on demand, but we also have a webinar section on our webpage. If you go to Learn and scroll down to Webinars, I list all the webinars that I've done on the website, so you can watch them on demand. But that effectively is just a link to the LabRoots page that you've used to come here. So yes, it will be available on our website, but it'll just be linking through to the LabRoots website you're already on. So it's best to watch it on demand from there, I'd say. But we have a long list of all of our webinars on there. So if you want to watch any of the ones that you missed previously, they're also all on there.
Wonderful. All in one place.
Exactly.
Perfect. And it looks like those are all the questions that we have time for today. Thank you again, Matt, for your time today and your very important research. We would also like to thank LabRoots and our sponsor, Teledyne Photometrics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and the LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your friends and colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you very much, everyone.